Hello, welcome back. In this section, we'll talk about types of inflammation. According to duration, inflammation can be classified as superacute inflammation, acute inflammation, subacute, and chronic inflammation. Superacute inflammation often lasts hours to days, and acute inflammation lasts days to 1 month. Subacute inflammation lasts 1 month to several months, and chronic inflammation is usually persists for more than 6 months. Chronic inflammation are associated with fibrosis and scar formation. According to the pathologic changes, inflammation can be classified into 3 types: ulcerative, exudative, and proliferative inflammation. Exudative inflammation, according to the exudate, can be classified into serous, fibrinous, suppurative, and hemorrhagic inflammation. Proliferative inflammation include general proliferative inflammation and granuloma. Now, let me introduce the first type, ulcerative inflammation. In ulcerative inflammation, ulcerative changes are the most obvious. Tissue injury and necrosis are predominant in the process, while exudative and proliferative changes are slighter. Ulcerative inflammation commonly seen in parenchymal organs. The common causes include virus, toxin, chemical poison, etc. Typical ulcerative inflammation include fulminant hepatitis, type B epidemic encephalitis, and caseous pneumonia. The two pictures show acute severe hepatitis. Lobular structure is destroyed with massive and severe hepatocyte necrosis. The liver is significantly shrunken and deformed. This picture shows type B epidemic encephalitis. Under microscope, here we find the pale staining softening foci. The brain cells died and were digested. The second type is exudative inflammation. Exudation is the most predominant features, while ulcerative and proliferative changes are slighter. Excess of one or other component of the inflammatory exudates results in distinctive morphology features of inflammation, including serous inflammation, fibrinous inflammation, suppurative inflammation, and hemorrhagic inflammation. Serous inflammation, as the name implies, the exudate derived from blood or serosal lining cells, is watery with a low protein content. Serous inflammation is commonly seen in mucosa, serosa, lung, and loose connective tissue and skin. For example, the running nose of the common cold and the cutaneous blister that occurs after a burn. The two pictures show the skin blisters result from a burn or scar. This picture shows the skin blisters result from herpes virus infection. Low power view of a skin blister showing the epidermis separated from the dermis by a focal accumulation of serous exudation. A small amount of serous exudation can be absorbed. However, severe edema in throat can cause suffocation. Severe edema in pleural cavity or pericardial cavity can cause dysfunction. Fibrinous inflammation occurs as a consequence of more severe injuries, resulting in greater vascular permeability. So, large molecules fibrinogen leaks into the tissue. Fibrinogen is converted into fibrin. The common causes include shigellosis, streptococcus pneumonia, Corynebacterium diphtheria. The lesions contain a large amount of fibrin, necrotic debris, neutrophils, and other cells. Fibrin accumulates and appears as an eosinophilic measurement of stress, or sometimes as an amorphous coagulum. Fibrinous inflammation is commonly seen in mucous membranes, serosa, and in the lung, and is observed in rheumatic pericarditis, dysentery, diphtheria, lobular pneumonia, etc. Fibrinous inflammation is commonly seen in mucous membranes, such as upper respiratory tract and intestinal tract. The main feature is the formation of cytomembranes. Therefore, this kind of disease is called cytomembranous inflammation. Fibrin, necrotic tissue, and neutrophils, bacteria, are mixed and form gray-white membranous substances, and cover the surface of organ, for example, bacillary dysentery, diphtheria. This picture shows diphtheria of pharynx and trachea. The mucosal surface of trachea and bronchi is covered with a thick and gray-white membranous substances. The exudate is called cytomembrane. At a low power, the mucosa shows degeneration necrosis with fibrinous exudates. There is a thick layer of cytomembrane covered on the surface of mucosa. At the high power, we can observe that cytomembrane is composed of fibrin, necrotic tissue, and neutrophils. The eosinophilic measurement of threads or amorphous coagulums are fibrin. This is a gross specimen of acute bacillary dysentery. The chronic mucosa shows erythema, ulceration, and cytomembrane formation. The cytomembrane is uneven, dirty, gray, and somewhere has fall off locally. At the low power, this is the intestinal wall. This is the mucosa, and here is the submucosa. And there is a thick cytomembrane covered on the surface of mucosa. Cytomembrane is composed of necrotic tissue, fibrins, and neutrophils. Fibrin may deposit on the pleural, pericardium, and peritoneum. Fibrinous inflammation is observed in rheumatic pericarditis. The fibrinous pericarditis is called hair heart. Fibrin deposits on the pericardium. which gives a shaggy appearance, is formed by the rubbing of the fibrinous exudate during the moment of cardiac pulsation. This picture shows a hairy heart with a shaggy appearance. This picture shows the gross view of lobar pneumonia with gray hepatization. The lower lobe is uniformly consolidated. The lung is dry, gray-white, and firm. Under microscope, we can see here is an alveoli, and the alveoli is filled with exudates, meaning neutrophils and fibrin, and also we can find some red blood cells. The picture shows a pink meshwork of fibrin exudate in the alveoli. Fibrin exudates may be degraded by fibrinolysis, and the accumulated debris may be removed by macrophages, resulting in restoration of the normal tissue structure. However, intensive fibrin-rich exudates may not be completely removed and are replaced by an ingrowth of fibroblasts and blood vessels. That's called organization, leading ultimately to scarring that may have significant clinical consequences. This picture shows advanced organizing pneumonia, featuring transformation of exudates to fibromyxoid masses, richly infiltrated by macrophages and fibroblasts. Organization of a fibrinous pericardial exudate forms dense fibrous scar tissue that bridges or obliterates the pericardial space and restricts myocardial function. Circulative inflammation is manifested by the presence of large amounts of neutrophils exudate, accompanied with tissue necrosis, pyogenic bacteria, and the formation of pus. The inflammation was usually caused by pyogenic bacterial infection. There are several variations of this type of inflammation, include abscess, phlegmonous inflammation, surface purulent inflammation, and empyema. An abscess is a localized collection of pus in an organ or tissue. Pus is a purulent exudate, contains neutrophils, pyocytes, necrotic cells, and pyogenic bacteria. Pyocytes are the necrotic neutrophils or degenerative neutrophils. This is a furuncle, an abscess of skin. Several furuncles can communicate to each other and form a carbuncle. Small abscesses may be absorbed. Large abscesses need surgical resection. As time passes, the abscess may become completely walled off and eventually be replaced by connective tissue. Because of the underlying tissue destruction, the usual outcome with abscess formation is scarring. Staphylococcus aureus may spread to other organs and form metastatic abscess. The picture shows cerebral abscess. On the cut section of the brain, there is an abscess with a clear edge, enveloped with a layer of fibrin capsule. The cavity of the abscess is filled with the yellowish or gray-red pus. This picture shows liver abscess. At the low power, a well-defined round foci can be seen in the liver tissue. This is an abscess. In the abscess, there are large amounts of neutrophils, exudates, and tissue necrosis. This picture shows a well-defined round abscess in the lung. The abscess contains neutrophils, cell debris, and bacteria, and is surrounded by congested blood vessels. This picture shows multi-abscesses in kidney. We can find many yellow abscesses scattered on the section of the kidney. Abscess may lead to a localized defect, inflammation of the skin and the mucous membranes with superficial necrosis, and detachment can lead to local defects, resulting in ulcer, sinus, and fistula. An ulcer is a local defect of the surface of an organ or tissue that is produced by necrosis of cells and shedding of necrotic and inflammatory tissue. This picture shows a chronic gastric ulcer, crater with an acute inflammatory exudate in the base. And as we can see here, the wall of the stomach is defective, forming an ulcer. Sinus is an abnormal tract from a cavity to the surface. Fistula is an abnormal tract open at both ends, containing two surfaces. Phlegmonous inflammation. This denotes widespread purulent inflammation in loose tissue caused usually by hemolytic streptococci, for example, acute appendicitis. The two pictures show phlegmonous appendicitis. The appendix was swollen, and the blood vessels are dilated and hyperemic. Then, look at this appendix. There is a layer of gray suppurative exudates on the surface. Under microscope, we can see here is the wall of the appendix, and the lumen of the appendix is filled with pus. And some area of the mucosa shows erosion. The mucosa shows erosion and extensive neutrophils infiltration. The lumen of the appendix is filled with pus. At high magnification, we can observe the pus is composed of neutrophils and debris and some fibrin. These cells are neutrophils. This picture shows the muscular layer of the appendix. These cells are smooth muscle cells. The intermuscular spaces become widened because of edema, and among the smooth muscle cells, we can find many neutrophils. It's important to distinguish between abscess and phlegmonous inflammation. Both are purulent inflammation. but there are many differences between them. The main differences is that the abscess is a localized purulent inflammation, and the phlegmonous inflammation is a diffuse purulent inflammation. Surface purulent inflammation is a purulent inflammation of mucosa or serous surface. An empyema is a connection or pus in a hollow viscus, such as the gallbladder or appendix. The term is also used for a connection or pus in the pleural cavity. The two gross specimens are purulent meningitis, a thick layer of supuratrial exudate filled subarachnoid space and covers the sulci and gyri, and meningeal vessels dilate and become hyperemic. Under microscope, a neutrophilic exudate is seen involving the meninges at the left, and the blood vessels are dilated, and there is edema and focal inflammation in the cortex. Hemorrhagic inflammation refers to vascular walls severely injured with exudate contained large amounts of red blood cells, inflammatory foci. The inflammation is caused by highly virulent organisms, such as epidemic hemorrhagic fever. So we have talked about ulcerative inflammation and exudative inflammation. Then we will talk about proliferative inflammation. In this part, we'll talk about general proliferative inflammation and granuloma, inflammatory polyps, and inflammatory cyst tumor. During the healing process, active cellular proliferation occurs. The dominant cells in this process are macrophages, endothelium, epithelium, and fibroblasts. Should the condition become chronic, lymphocytes, plasma cells may predominate. The proliferation of cells can result in characteristic morphologic appearance at the inflammatory site. Under microscope, we can see in the region narrow meaning macrophages and meaning lymphocytes. The second type is granuloma. The granuloma is a larger lesion composed of macrophages, surrounded by a collar of lymphocytes, plasma cells, and fibroblasts. There are two types, infective granuloma and foreign body granuloma. Infective granulomas are observed in tuberculosis, syphilis, fungal infections, cat scratch fever, etc. Granuloma is a larger lesion composed of macrophages, surrounded by a collar of lymphocytes, plasma cells, and fibroblasts. This is a typical granuloma of tuberculosis. This is a low-power microscopic view of tuberculosis. We can see several granulomas with clear border scattered in the lung. In the center of granuloma, there is caseous necrosis. At high magnification, the necrotic tissue is amorphous, granular, and pink. The details of the cells are not clear, and the structure of the tissue is completely lost. Here is caseous necrosis, and around the caseous necrosis, there are many epithelioid cells. These cells are epithelioid cells. They are derived from macrophages. They quite resemble epithelial cells, so we call them epithelioid cells. They are large. The boundary is not clear. The nucleus is large, round, or oval, and the staining is very light. The cytoplasm is light and rich. The two cells are Langhans cells. They derived from the fusion of multiple activated macrophages. Langhans cells have large cell body and multiple nuclei. The nuclei arranged in garland or horse's hoof form or assembled together and quite resemble those of epithelioid cells. The cytoplasm is red stained and abundant. The picture shows tuberculosis of lung. The two cells are Langhans cells. Foreign body granulomas, as the name indicates, these granulomas form in response to foreign bodies, for example, splinters, sutures. The region is composed of foreign bodies, foreign body germ cells, and epithelioid cells. Foreign body germ cells formed when macrophages phagocytize insoluble material or particles. Nuclei are scattered throughout the cytoplasm. No caseous necrosis was seen. This picture shows a granuloma in response to lipid cholecteral crystal. Here we can see many cholecteral crystals. This picture shows a foreign body granuloma caused by sutures. Two foreign body germ cells are seen just to the right of center, where there is a bullish strand of suture material from the previous operation. This picture shows a piece of plant accidentally inhaling into the lungs, causing granulomatous inflammation. This is a foreign body germ cells. It's huge, and the nuclei are arranged in disorder. Inflammatory polyps are small growths that look like warts. This picture shows two polyps of vocal cord. Inflammatory cyst tumor. It's a tumor-like mass pathologically composed of lymphocytes, plasma cells, macrophages, and foam cells within a spindle-shaped stroma. That's all for this section.